So we're talking now about mechanisms of infectious disease. And with this, we're going to talk about agents of the infectious disease, the epidemiology of it, how is the mechanism of infection, what are the modes of transmission, what symptoms can you see, how is the site of infection is going to look, how does an infectious agent is classified as a virulent versus non-virulent, and then some of the diagnostic tests and treatments, and we're going to talk a little bit about the bioterrorism and pandemics. Go figure. So some of the terminology you're gonna get used to when you're talking about infections, like the host. So host is anyone that will embrace the organism and make it its place to grow. And the infectious disease is the disease that's brought by the fact that the host is now surviving and thriving in the new organism. And then the colonization versus infection is just the presence of the living organism within the host, not necessarily causing an infection. And so many times you're going to see just pure colonization of an infectious agent. Microflora is and you're gonna, or you hear it called the normal flora. It's the bacteria that lives on the surfaces of the body. Virulence is the ability of that virus or bacteria to cause a disease. Pathogens are those microorganisms, just another name for them. Mutualism is the ability of the microorganism is to the to exchange, favors kind of thing. These are some. When we talk about the infectious disease process, you're gonna hear a couple of places. You're gonna hear them talking about different things. You may hear triad of infectious disease model, which is what your book talks about, or you hear the chain of infection model, where you see the infection, there's the host, there's the portal of entry, there's the organism portal of exit, reservoir, and you see the vulnerable person that's going to get disease. In this model, they're talking about you're going to have an agent, you're going to have a host, you're going to have environment, and there's always that reaction and the circle going around where the disease can form or not. And we saw that in COVID, like people who were at risk for, their environment and their vulnerability affected whether they were diseased and symptomatic, or they were diseased and asymptomatic, or whether they did not capture the disease. And some of the infectious diseases, you need to know them, the agents, I mean, for infectious diseases. You have the bacteria, everybody knows it, viruses now on every mouth, fungi, parasites, prions are one kind of like the way the toxins they produce cannot be killed regularly. And these are the two differentiating agents we'll talk about in a minute. Um, Viruses, they're small, they don't have a structure. This is if we ask questions, we're going to ask questions about this. They either have a DNA, RNA, they will replicate plentiful, but it has to be within a living cell. That is very much how viruses live. Bacteria, you have two type of bacteria. They're either gram-positive, that means when you put them on the agar plate, they're going to be staining purple, and the gram-negative, that will not be stained. The gram-negative usually are your worst organisms, and they have their own antibiotic. Gram-positive, they have their own antibiotics. And then you have a third family of antibiotics that can work for both. These Rickettsia, Anaplasmataceae, and you don't have to say the names. They're just organisms that have both viral and bacterial characteristics. Just know that of them. And then fungi, yeasts, mold, these are the fungi. The parasites, all those protozoas, helminths, arthropods, and um, there's that method of transmission from animals and from vectors to the humans. So if we want to say which of the following pathogens does not fit the typical description of an organism, you're gonna say virus. Why? Because it does not have an organized cellular structure. Makes sense? And then we go back to epidemiology. We covered this earlier in the course, and we said that epidemiology is the study of factors, events that influence the transmission of diseases. In this particular case, we're studying more the infectious disease. And again, same like we talked before, incidence is going to be the incidence of infectious disease, and that's how many cases of an infectious disease have occurred within a defined population, and prevalence is how many active cases we have at any time, and that's what they're counting every day for COVID, if you care to forget, because we can't forget, because we have to hear those numbers every day. And then endemic versus epidemic versus pandemic. We covered this. Endemic is an infectious disease that is prevalent in this specific geographic population. For example, malaria is in certain areas of Africa. The Zika virus was found in South America. Epidemic is an abrupt increase in cases, such as what happens to us with the flu virus. Pandemic is COVID. Yeah, COVID pandemic is going to be the synonym of it for life. And then when we talk about modes of transmission, you've heard of contact, airborne, droplet. It's kind of the same, but they're here taking it to another level where you're talking of more scientifically in pathophysiology. Penetration, so it's that way of how it enters. It has to enter in. The ingestion is like you have to eat it. Inhalation, you just inhale it. And thus, you're going to hear some of the protection is by stopping that mode of transmission. They call it transmission-based precautions. And there's one interesting thing that we have to talk about is where did that infection come from? If it came from within a hospital, they call nosocomial. The newest word is hospital acquired or healthcare acquired, because not always it's a hospital, sometimes it's an outpatient, versus a community acquired outside of a healthcare facility. And then host, we talked about what host means. And then the endogenous versus exogenous, that's when we talk about symptoms, there are symptoms that are going to be specific. We ate contaminated food, we're going to have diarrhea, or versus non-specific, having fever, malaise. Look at the loss of taste or smell and COVID disease. And sometimes it's going to be obvious. We know that we get the chickenpox, we're going to have some signs of symptoms of flu-like, then the rash comes out and stuff like that. So you can predict how the symptoms gonna evolve in certain diseases. And then again, we're talking about those course or the way the disease is gonna progress. And so you talk about the incubation period. That's the time that the organism is building, and we talked about those stages in the first lecture. It's going to be the same in the disease in general versus an infectious disease in specific. Um, what is going to influence the infectious disease is what type of pathogen. It's very important. Where did it come to? Is it in our urine? Is it in our lungs? And how that host is immunologically prepared to fight. So, is this statement true or false? uh, the infectious symptoms are always going to be obvious and apparent. No, sometimes they're covert, they're not specific. And then, so we talked about how that my organism is acting, the virulence factor of it. There are organisms that are toxin producing. The more they're under toxin, it's worse than exotoxin. The way they adhere and the way they're formulated is going to affect their virulence. When we talk about diagnosing of an infectious disease, it is important to be able to know what was the probable pathogen. Like in the case of COVID, they didn't know it. It's a novel uh, uh, virus, so it took a while to recover what is the probable pathogen, and that is essential. And so and another thing that goes hand in hand with ability of taking and getting that probable pathogen is to be able to accurately identify the clinical signs and symptoms with that infectious disease process. And we also saw that in COVID, like the loss of smell and loss of taste was identified later on, which was left so many people not identified. So what do you do to test? Some of the lab diagnostic tests is to do culture. So you take the urine and culture it, you take the blood and culture it, you take the stool and culture it. And also you take some blood results, because blood results, especially white blood cells, especially the CBC, is going to reveal very much about the infectious status of a person. Of course, if we're looking at a virus, we're going to look at the DNA, RNA sequencing of it. Makes sense? And then when we treat, it goes without saying that if the patient is growing a bacterial infection, we can't give an antiviral agent. And same wise, you go to your doctor and they say you have the flu, you have influenza A, and you want an antibiotic. Well, it's not going to work. The only thing that's going to work is the antiviral agent. And then when they talk about antibiotic, you're going to hear the word bactericidal versus bacteriostatic. And as the name implies, bactericidal will kill, it's lethal and it will kill the pathogen, versus bacteriostatic is just going to put it at bay, it's going to put it to sleep. So how does it kill it? There are certain types of antibacterial, antimicrobial that will interfere with the cell wall. Some will interfere with the bacterial protein synthesis. Some will interfere in the nucleic acid synthesis. Some will interfere in the bacterial metabolism. So that's how different antibiotics work, or antimicrobial work differently. For example, penicillin will work on the cell wall, same as the cephalosporin and the monobactams, versus the aminoglycosides, the tetracyclines, the macrolides, they all work on the ribosomes. Sulfonamides work on the folic acid, and quinolones work on the DNA synthesis. So you see how different families of antibacterial agents work differently on the bacteria. You may see also that the host immune response may be boosted with given immunoglobulin therapy and such, so that it is able to fight and naturally produce that inflammatory phase and giving that phagocytosis so that it can fight that infectious agent. Um, Non-pharmacologically, sometimes you need to debride the area, remove it. For example, if the appendix is inflamed, infected, you just go ahead and do appendectomy. That's the only way you can get rid of that infection, through surgical interventions. So, is surgical therapy used in tandem with antibiotic in some cases of severe infection? Yeah, we sometimes need to debride the area in addition to giving infection antibiotics, so that the recovery process is enhanced. Sometimes we just do the surgical process to drain the abscess, not always to remove the organ. So then bioterrorism goes hand in hand here with infectious disease, because usually the bioterrorism agents, they're infectious agents such as the plague, the smallpox, or viral encephalitis. And so they categorize them: category A, category B, category C. And of course, when we think of global infectious disease, in addition to all those diseases that we've known for a while, Zika just was very famous maybe a couple of years ago. It still probably is active in places of low income, but SARS coronavirus number 2 is the new virus from like the mutating from SARS, and so it is a pandemic. So, and it is a global infectious disease that has come to life in 2018. And with that, we'll conclude our lecture.